Okay, so welcome back to my office. We're going to talk about nucleic acids now. Oops, let me pull my thing back up and share my screen with you guys. Okay, so some would argue that nucleic acids are the most important of all our four kinds of biomolecules, although I think they're all pretty important. We wouldn't be able to live without any of them. But the nucleic acid is the genetic code. So there's two kinds of nucleic acids, DNA and RNA. So there you go. That's the answer to the first question there. But also what I want you guys to be able to tell me is, how are DNA and RNA similar and different? So this is a nice little Venn diagram type situation. And then what are the roles of DNA and RNA in our cellular processes and function? Okay, so your book refers to the genetic code as a blueprint, but I think that that is not an accurate analogy. I like the analogy of a recipe much better, because in a blueprint you can look at the drawing and say, okay, oh yeah, here's the window, and there's the door. But in a recipe, the words and the information that are on the card are not a direct one for one like we see in a blueprint. If I give you my grandma's recipe, you might make that lemon meringue pie very different than my grandma makes it, because you might have different measuring cups, you might be doing it at a different temperature, you might be beating your eggs a little bit longer or a little bit shorter, and so we might see these minute differences. And I think that's a better analogy for what genes are, because there's not genetic determinism. If you have a gene for something, that doesn't necessarily mean that you're going to express that trait. Your environment also influences how your genes are expressed. So that's why I like the analogy of a recipe better than a blueprint. All right, get off my soapbox, and here we go. So deoxyribonucleic acid, yeah, DNA. This is one of the check boxes that you have to check in order for you to be considered alive. If you don't have DNA, you're not alive. Viruses, most of them only have RNA. Some of them have DNA, but they're still not alive because they're not cellular. All right, but in eukaryotes like me and plants and fungi and other types of animals, our DNA is stored in a membrane-bound nucleus in our cells. In prokaryotes, there's just this one kind of circular chromosome that's just like chilling out in the cytoplasm, which I like to call cell juice. Chloroplasts and mitochondria of eukaryotic organisms also have DNA that's similar in structure to prokaryotes. So every mitochondria inside your body also has one chromosome in a circle, just like prokaryotic organisms. I wonder why. All the genetic materials in a cell is referred to as the genome. That's all that genetic information. And then the study of genomes is called genomics. Okay, so in eukaryotic organisms like us, like fungi, like plants, the DNA molecules, so here's our little double helix that we know and love, interact with these proteins called histones. And then that kind of the DNA molecule winds up around those histone proteins to form chromatin, which is going to eventually, once we get a bunch of it, that's going to form our chromosomes. And so we have this kind of a nice little compact storage unit for our DNA. So that becomes really important. And this is only found in eukaryotes. Okay. So in our RNA, our ribonucleic acid, its main job is to synthesize proteins. And there's lots of different kinds of RNA that have different jobs. The most common that we see is mRNA, or messenger RNA. So we give it a little lowercase m, uppercase R, uppercase N, uppercase A. And so in this diagram, we have this little light blue circle-ish kind of thing that's supposed to represent our cell. And then we have a little darker blue circle representing the nucleus. In the nucleus, we have the DNA. We're going to make a cheaper copy of that DNA because DNA is obviously very valuable. That's our original copy. And then we make a cheaper copy called RNA. And then this messenger RNA actually is the thing that leaves the nucleus to go out into the cytoplasm or the cell juice and to create the proteins. Okay, so that was the quick and dirty overview of DNA and RNA. Let's talk about the structure of these things. Our monomers are our nucleotides. Nucleotides have 3 components to them. We have a 5-ring sugar. That's called our pentose sugar. So here, that's where that is. And then hooked onto one of the carbons, carbon 5. Remember our oxygen atom, we started saying, okay, here's carbon 1, here's carbon 2, here's carbon 3. Carbon 3, that's an important one. Carbon 4, and now we're going out of the ring up to carbon 5, and that's where the phosphate group is. And remember that phosphate groups have these 2 negatively charged oxygen atoms, and that negative charge is really important for how that thing is going to interact with our aqueous environments inside our bodies. Okay, so we got our sugar, we got our phosphate, and now we need a base. And so that can be what you might have heard referred to as our A's, C's, G's. And then if we're talking about RNA, we have a U instead of a T. And that is shown here attached to carbon 1. And right here we have one of our, that's our base. Okay. So the bases in DNA, I should change this. The bases in DNA are either classified as purines or pyrimidines. And it's just telling us, is there 1 ring in our pyrimidines, or are there 2 rings like we see in the purines? So adenine are A's, and guanine are G's. Our purines, they have 2 of these carbon rings. This nitrogen, that's what makes it a base. And then the pyrimidines, they just have the 1 ring, cytosine, thymine, and then uracil. So we'll get to that in just a minute. These are formed, our polymer of our nucleic acids. They're formed by our friend dehydration reaction. And so the bond occurs between the 3' carbon, and that's what this means. 3, and then this little apostrophe means where you say that as prime carbon of this dude. So here's our oxygen, 1, 2, 3. Okay, it's going to link with the phosphate group that's attached to the carbon 5 of the next monomer. So this is how we're linking those Legos together. The 3' carbon hooks up to the phosphate group, which is attached to the 5' carbon. And so when we talk about DNA replications, we're going to talk a lot about 3' to 5'. And that's what we're meaning, is that order of how these Lego blocks, in our analogy, are linking together. Because like if you think of 2 Legos and there's like the flat end and then the end with the pokey ups, you can't link them together when the 2 flat ends together. You have to link them up. You got to link them up like that. And that's the same for our nucleotides, is that they have to be linked together in a specific way, and that's from 3' to 5'. So the structure of DNA, once it's kind of built, it starts to form this double helix. And so you can kind of think of it as a ladder that's been twisted. And so here is the phosphate sugar backbone. It kind of goes around in this little like twist, and then the bases would be the rungs of the ladder. And those are held together through hydrogen bonding. And now what you guys will notice if we look at adenine and thymine is that there's 2 links for them to create these hydrogen bonds. But if we look at guanine and cysteine, then there's 3 links. Oops. I gotta change this. But so that's why we often see this pattern of pairing. We see that, I'm going to change it right now because that's going to bother me. Live modifications to the PowerPoint. Oops. Okay. So our adenine and our thymine are held together through 2 hydrogen bonding links, and then guanine and cysteine are held together by 3. So that's what those dashes, those hyphens are there. So remember that individually the hydrogen bonds are weak, but you can see that there are a huge number. And so it does take a lot of energy to untwist this ladder and to break these DNA molecules into its 2 single strands. When I do this in my research lab, we have to heat up our solution that has our DNA in it. We have to heat it up to 95 degrees Celsius, which is just below boiling, in order for it to uncoil. So if a certain DNA molecule has bases made of 30% thiamine, can we figure out how much cysteine it has? And I'm going to stop sharing my screen for a second so I can write it out on the whiteboard. So we know, well, let me focus myself back. Nope, it's worse. Okay, so we said that thiamine has 30%. Okay. Ooh, that's backwards. Okay, I'll just do it here. Share my screen again. Okay. Ooh. Okay. Where are we? Here. Okay. So we know that A's and T's always match up with each other, right? So if there's 30% T's, we know that there has to be also 30% A's. So now if we say, okay, we know that we have 30% A, 30% T, add those together, that's 60%. That leaves us with 40% for everything else. And we know that C's and G's match up, and so there's going to be about equal amounts. So we can take that 40% and say 40% are 20% are C's and 20% are G's. So make sure that this makes sense to you and that you can do these calculations because this might be on the exam. Okay. So if I give you any percent of one of the bases, you can figure out what the percentage is for the rest of them by knowing the A's and T's go together and C's and G's go together. Okay. And then there's always a complement to the DNA strands. So if we know that A's always pair with T's, and so then T's always pair with A's, we could write out the complements of each of these. And so if that is unclear to you on how to do that, please talk to me as soon as you can. We can do a little Zoom conference or whatever to make sure that you understand this, because that's important. Okay, and so DNA in our double helix form is, we call it anti-parallel. So going from our 3' to 5' ends, so our Lego blocks are attaching in this direction. And then the other strand, they're coming whoop the other way. So this arrow coming down from copying, going from 3' to 5'. So they're anti-parallel. So this is important when we start talking about DNA replication, because what we'll find is that because they're anti-parallel, there's all these weird little things that happen with the guy going the wrong way. Okay. We'll get into that later on when we talk about DNA replication, but also know that they're anti-parallel. So they're kind of pointing in opposite directions. Okay, so now let's talk about our buddy RNA. So how does ribonucleic acid differ, and how is it similar to deoxyribose nucleic acid? Well, obviously they have a different sugar, right? RNA has the sugar ribose, whereas DNA has the sugar deoxyribose. And just remember that on that carbon 2, there's one less oxygen atom on the deoxyribose. It's been deoxygenated a little bit, so that's why it's called that. RNA uses the base uracil instead of thiamine, but the other 3 bases are the same between DNA and RNA. RNA is usually found in just a single strand, whereas our DNA is found in this double helix thing. However, just because RNA is found as a single strand, it also folds in on itself in these predictable patterns that we can describe. RNA, like I said at the top, it has lots of different kinds of jobs, and we'll kind of go quickly through each of these. So I already talked about messenger RNA, mRNA a little bit. That kind of stuff has been in the news lately. So now you can go and tell your family about this and why COVID vaccines can't change your genetic material, because that's not what RNA does. And then we also have ribosomal RNA, which we'll talk about. It's important for protein synthesis. Transfer RNA are little short pieces of RNA that go and fetch the amino acids during protein synthesis, and microRNA that kind of just like is the supervisor of all that stuff of protein synthesis. Okay, so mRNA carries info contained in DNA out into it leaves the nucleus and takes that information out into the cytoplasm. mRNA is the complement of DNA, and so remember, this is going to get this is going to become important when we talk about protein synthesis, and we have a little lab about protein synthesis, that you got to make sure that you're talking about the complement. DNA. As previously mentioned, this is what leaves the nucleus, and it interacts with the ribosomes. Ribosomes are the organelles where protein synthesis happens, and it can interact with some other organelles as well. And so here's just a little figure of our mRNA, and you can see it's kind of broken into these different colors, and that is important, and we'll talk about that soon. And then we've got our tRNA here and our protein that's being built. Okay. So make sure that if you said, if I gave you this sequence of DNA, that you could tell me the mRNA sequence. And the tricky part is that, so T's always match with A's. T's always match with A's. A's match with T's in DNA, but in RNA, we give it a U because it has that uracil instead of the thymine. So make sure that whenever you are thinking, okay, I'm in DNA or I'm in RNA land, here I am talking about RNA, I'm going to think uracil instead of thymine. Okay, that's where students get hung up a lot. So make sure that that's clear to you. And if it's not, you can come talk to me because my job is to help you know this stuff. Okay, so protein synthesis. Whoo, boy. Okay, so the mRNA is read by ribosomes, and those are those important organelles used in protein synthesis. And ribosomes read the mRNA in these 3 bases blocks, and we call those codons. Each codon codes for a single amino acid. So as the protein's being built, the sequence of the bases in the RNA tells the tRNA what amino acid to go fetch. Go get me a valine. Go get me an adenine, right? So these are different amino acids that are being called for in our recipe. Okay. RNA, the ribonucleic acid, this just lines up the mRNA with the ribosome. So if you guys kind of think of like the dude at the car wash, where you drive up and then he's like, and you have to kind of turn your wheel so that your wheel lines up with a little track that takes your car through the automatic car wash. It's kind of RNA's job is to be like, no, over here, okay, all right, now you're good to go. Take your foot off the brake, put your car in neutral, enjoy your car wash. Okay, and it also catalyzes the peptide bond formation between those amino acids. So it's catalyzing, it's reducing the amount of energy needed to do that dehydration reaction to link up those amino acids. Concepts are starting to come together. Yes. Okay, tRNA, again, these are little short guys. They're usually only like 70 to 90 bases long, so they're fairly short. And again, their job is to go and fetch the correct amino acids to link into that growing polypeptide chain. Okay, this is how the primary structure of our proteins is synthesized, is based on the information that was contained in the DNA, transferred to the mRNA, and now the tRNAs are the kids that go and fetch the ingredients to build the protein. MicroRNA, these guys are super tiny and they just help regulate protein synthesis. So that's kind of their jam. They're just really little, and they're kind of the helpers. And so that's kind of all we'll say about those guys for now. So now we've kind of described the central dogma of biology without being explicit about it. So now let's be explicit. So the central dogma of biology says that DNA replication happens within the nucleus. DNA is copied. That information is transcribed onto RNA and then translated to a protein. Now, getting the order, because transcription and translation are very similar words, and so it's easy to get them mixed up. And the way that I remember them is that we have, so in the nucleus is our master copy of our genetic code, encoded in our DNA. The analogy that I like to use for this is when I was working at the U.S. Geological Survey, one of my jobs was to write about the 150th anniversary of John Wesley Powell going through the Grand Canyon. So here we got our first white dude exploring the Colorado River, and it also kind of lined up in the same summer as the 50th anniversary of the moon landing. So that was kind of cool. But so when I was writing this, when I was working for the USGS, I learned that NAU houses John Wesley Powell's original journals that he wrote as he was traveling down the Colorado River through the Grand Canyon. And I was like, dude, I need to go check that out because that's really cool. And so I got to the library and they were like, okay, yeah, we're gonna like not let you look at those because they're really important. If you want to look at them, you have to like go through all the safety things. You have to wear gloves. You have to like leave all your stuff out here. You can't take your coffee in there because that original copy was so valuable. And I was like, dude, that sounds like a lot of work. I don't really want to do that. So I went online and I found a cheap copy um, of John Wesley Powell's journals and the journals of all the people on the trips with him. And then I could get that same information without the risk of endangering the original copy. And so that's my analogy for the DNA. That stuff's too valuable. You don't want that to get messed up, because if that gets messed up, you're in big trouble. But you can make a copy of it that's cheap, that's easily broken down, put back together, and if that gets damaged, then who cares, because you can just make another copy. Now, the analogy from going from RNA to proteins, or translation, that process, that one's an easy one to remember because you're going from nucleic acid language with DNA and RNA, but now you got to translate it into protein language. And so if we use the John Wesley Powell analogy, I speak English, so I don't need John Wesley Powell's English writings to be translated. But if I was somebody that spoke only French or only Italian or any other language, then that John Wesley Powell's journals would need to be transcribed into a cheaper copy, that if that gets messed up, then it doesn't matter. But it also would need to be translated into a language that I could understand. So transcription's going from nucleic acid language to nucleic acid language. Translation is going from nucleic acid language to protein language. So make sure that you understand this, because this trips up a lot of students. But transcription and translation are related but different. And it's really important to know what is happening to those genes, because if a gene is transcribed but not translated, then it doesn't really matter if there's mutations occurring in that transcription, because there's no proteins made from it. So who cares? And I use that in my research. One of the cool things about fungi is that they have an area of their genome it's really important. It's actually coding for the proteins that become ribosomes. And so there's a strong evolutionary pressure to not mess that up. You don't want mutations in there, because if you can't make proteins, you're toast. So But it's also, so we've got these nice little conserved regions where there's not very many mutations. And then right next to it, there's a place in the genome that's transcribed but not translated. So if the mutations occur there, it doesn't matter. And that works great for me because now I know how to build a primer so that I can say, okay, start your DNA sequencing here, latch on here, okay, copy, 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 copy into this variable region where mutations do occur. And then I can say, okay, now I know what species that is, even though I don't have a mushroom in my hand. If I was able to pull fungal DNA out of a plant, I can say I know what species of fungus that is because I looked at its genome in this place where it was transcribed but not translated into proteins. Okay, so make sure that you understand the central dogma: transcription, translation, how they're similar, how they're different. And then here's a nice little summary table for you looking at DNA versus RNA. And so also get to know this, be able to draw me a nice little Venn diagram of the similarities and differences in DNA and RNA. Okay, so do this again. What are the 2 or 3 main takeaways that you think that I want you to know for the exam? And then if you have a couple of 2 or 3 questions that you would still like answered. Okay, I will see you guys on whenever I see you next. So I'm going to stop sharing and have a great day, and I'll talk to you guys soon.